Welcome to the 13th episode of Histo Helpers. In today's episode, we're going to be taking a more in-depth look at the skin. And by the end of today's lab, make sure you're able to identify the two primary skin layers and their respective constituents, tell the differences and similarities between thick and thin skin, and recognize some skin appendages that we will be identifying during this lab today. Let's jump right in with slide 232, which is a section of thick skin stained with H&E. Thick skin is only found in areas where there is a lot of abrasion, such as your fingertips, palms, and the soles of your feet. It has a thick epidermal layer and a thinner dermal layer, and does not contain hairs, sebaceous glands, or apical sweat glands. At the top you can see the epidermis, which is spanned by the green line, with the dermis just beneath it spanned by the blue line. Mammalian skin proper is only really composed of these two primary layers, the epidermis and dermis. The subcutaneous fascia, or hypodermis, is located beneath the skin and is seen spanned by the orange line. Let's now take a closer look at the epidermis. As you know, it is composed of stratified squamous keratinized epithelium and serves as a protective barrier over top the body's surfaces. It is responsible for preventing pathogens from entering, for regulating body temperature, and for maintaining proper hydration. In thick skin, it is composed of 5 basic layers of keratinocytes: the stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum on top. As the epidermal keratinocytes mature, they divide, differentiate, and migrate from the deepest layer, the stratum basale, and towards the most superficial layer, the stratum corneum. Along the way, they become keratinized, lose their nuclei, and die. It takes approximately 4 weeks for a cell to reach the externalmost layer, where it is then sloughed off. The stratum basale, or stratum germinativum, is the deepest layer and is indicated by the yellow arrow. These are the keratinocyte stem cells that replenish the epidermal epithelial cells as they migrate to the exterior. Next, we have the stratum spinosum, which is spanned by the pink line. These large polygonal cells have lighter staining nuclei that are actively synthesizing components of the desmosomes that interlink the keratinocytes, which serve as strong connections between cells. If you look closely, these cells get their name from the spiny appearance of their characteristic desmosomal junctions. The next layer is the stratum granulosum, which is indicated by the red arrow. These cells are actively synthesizing keratohyalin, which is stored in the granules that are visible under light microscopy. Keratohyalin and other proteins synthesized during this stage promote dehydration, aggregation, and formation of an impermeable lipid-containing epidermal membrane that gives the skin its homeostatic functionality once it reaches the stratum corneum layer. The stratum lucidum, indicated by the green arrow, is the next layer and is only present in thick skin. It is characterized by the thin, lighter staining layer of the epidermis composed of dead keratinocytes. The most superficial layer is the stratum corneum, which is seen spanned by the white line. It is characteristically thick in the thick skin and is composed of dead, denucleated, keratinized epithelial cells that shed once they reach their most superficial layer. Now let's take a look at the interface between the epidermis and dermis. These invaginations of the dermis are called dermal ridges, such as that indicated by the yellow line. These dermal ridges are what give you the unique pattern on your fingertips and palms of your hands. You will commonly see capillaries of the superficial dermal plexus, such as those indicated by the red arrows, traveling up into the dermal ridges. You may also see Meissner's corpuscles extending up into the dermal ridge, which have an appearance like the stack of pancakes you had for Mother's Day brunch back in '08. Here is a good example of one indicated by the green arrow. Please do not confuse these with capillaries, like Brock did on last year's histology exam. Meissner's corpuscles are mechanoreceptors that are responsible for light touch, which is why they are more commonly seen in the dermal ridges of your fingertips. The dermis, which was discussed in depth in the connective tissue lab, is composed of 2 distinct layers. The more superficial papillary layer is composed of loose connective tissue, with the deeper reticular layer composed of dense irregular connective tissue. The dermis is composed mostly of type 1 and type 3 collagen fibers, which give it its tensile properties, along with the elastic fibers, glycosaminoglycans, and resident connective tissue cells. Down here you can see components of an eccrine sweat gland, with a lighter stained secretory portion, indicated by the yellow arrows, and its darker staining duct, indicated by the green arrows. Surrounding the lighter staining secretory portion, you can see myoepithelial cells, indicated by the red arrow, which are epithelial cells that contain smooth muscle contractile properties and function to squeeze the fluid out of the acinus's lumen. If you scroll up, you can even see a duct extending up towards the surface of the skin. Eccrine sweat glands perform merocrine secretion composed of a watery isotonic solution, and are in high density where you have thick skin. Lastly, take note of the nerves, arteries, and veins located most prominently at the dermis-hypodermis interface. We're now taking a look at slide 6, which is a section of the fingertip stained with H&E. I'd like to point out a couple mechanoreceptors that were less evident on the previous slide. First, a Meissner's corpuscle in the dermal ridge is indicated by the brown arrow. As I previously mentioned, these receptors are responsible for light, discriminative touch, characteristic of your fingertips. Next, take a look at the deeper Pacinian corpuscle, which are the large lamellar mechanoreceptors indicated by the green arrows. Pacinian corpuscles are tactile mechanoreceptors responsible for deep pressure and vibration. We have now transitioned to slide 5, which is a section of hyperpigmented thin skin. Thin skin has a thin stratum corneum and does not contain a stratum lucidum. It also contains hair follicles, sebaceous glands, and apocrine sweat glands. Although the epidermis is thin, the dermal layer of thin skin is relatively thick. If you want, pause the video now and try to orient yourself by identifying the 4 layers of the thin skin epidermis. Although the vast majority of the epidermis is composed of keratinocytes, there are 3 other types of cells present: Merkel cells, Langerhans cells, and melanocytes. You can't see Merkel cells or Langerhans cells by way of normal histological staining technique, so we're going to focus on the melanocytes, which are indicated by the red arrows. These melanocytes, which are of neural crest origin, are located among the stratum basale and have dendritic processes that extend into the overlying stratum spinosum and surrounding stratum basale. Melanocytes are responsible for synthesizing melanin, which is packaged into melanosomes and delivered to surrounding keratinocytes via cytokine secretion. If you look closely, you can see how melanosomes are situated like a hood over top the keratinocyte nuclei to protect them from exposure to mutagenic UV rays. Let's transition to slide 3, which is a section of the scalp stained with H&E. As I zoom in, you can see a hair follicle stained in cross-section. The hair follicle is surrounded by a dermal or connective tissue sheath, which is spanned by the gray line. The external root sheath is spanned by the red line. The internal root sheath is spanned by the blue line. Finally, the cortex of the hair can be seen spanning the green line, while the medulla is indicated by the orange line. We're now looking at slide 234, which is a different section of thin skin. Here, you can see a longitudinal section of a hair follicle. The dermal sheath is indicated by the yellow arrow. The external root sheath can be seen spanning the red line, and the internal root sheath is spanning the green line. Lastly, the cortex is indicated by the blue line. Because none of these hairs were sectioned down the middle, you won't be able to visualize the medulla. The erector pili muscle, indicated by the orange arrow, is composed of a bundle of smooth muscle fibers that cause your hair to stand up when you're cold, aroused, or scared. these fibers insert into the connective tissue base of the follicle. Take a look at this sebaceous gland, which is indicated by the green arrow. Sebaceous glands secrete an oily substance called sebum into the hair follicle. This gland undergoes hologen secretion, which means that the secretions are released by the rupture of the cell's plasma membrane, resulting in the release of intracellular constituents into the follicle. Let's finish up by taking a look at slide 14, which is a section of the fetal finger stained with H&E. The nail itself is called the nail plate and is indicated by the blue arrow. The nail plate is directly overlying the nail bed, which is indicated by the black arrow. The proximal part of the nail bed is called the root, or germinal matrix of the nail. It is indicated by the red arrow, and is home to nerves, lymph, blood vessels, and stem cells that give rise to the nail plate itself. Since you're interested in learning, take a look at your fingernail. The white part under the base of the nail is a portion of the nail root called the lunula. The eponychium, which lies just proximal to the cuticle, is indicated by the green arrow. Lastly, the hyponychium is the junction between the distal free end of the nail and the fingertip, and is not well visualized here due to artifact. That's a wrap. I really enjoyed giving you the skinny on skin, and I hope you did too. Tune in next time for an absolutely breathtaking look at the respiratory system with our favorite histo helper, Elliot King. As always, thank you for watching Histo Helpers. We're here to help.
And me too.